Well, it's really a pleasure for me to be back in Conf 4. So this room has actually a lot of memories from some time ago. I would like to say I defended my PhD here, but it's actually not true because it was in renovation, as you can imagine. So it was somewhere over there. But it's really nice to be back here. So the work I would like to present to you today is actually work that we did over the last 10 years maybe in our group at the PMMH. And so it includes many people who have worked on that. So Benjamin and Peixin were two PhD students who just defended in the group. Gaspar contributed, Nuris, Francesca and Martina. I have a lot of collaborations of very close colleagues here at PMMH or close. So all of the work I present to you today was done in collaboration with Eric Clement, some of them with Olivia, and also some recent work with Teresa. And our lab works on experimental approaches. So we have also a number of collaborators that computed to the theory I will present to you today. And so the theory has been done by Arnold Mathiesen and Andreas Zöttl, and part of it by Rodrigo Soco. And somehow, I don't know why, I left out Guan Ying, who was a postdoc in the group and who is now back as an assistant professor in China. And I should point out before I go on that actually all the beautiful drawings that you will see have been done by Martina. So unfortunately, I myself cannot draw as nicely. So to give you a little bit of background, because as I understood from Christina, it's a broader seminar and not everybody is necessarily a specialist on active matter, I wanted to tell you first of all, what is our definition, what do we understand, what is active matter. So actually active fluids, well, and first of all, I'm not talking about everything in active matter, but I will talk about active fluids. And so what we understand about an active fluid is that it's a suspension of particles, but these particles are self-propelled. And self-propelled means that they locally consume energy which propulses them. So applying a magnetic field from the outside would not create an active particle. And the fact that they consume energy at the scale of the particle means that these fluids are inherently out of equilibrium. And this actually converts them many interesting properties. So, for example, they can generate flows without external gradients. So here I will show you two examples. So the first example is from Denis Barthelot's group in Lyon. It's Quinker rollers. So it's particles that autopropulse themselves well, through the application of an electric field. And it's like on a microfluidic racetrack. So what you can see that after some time there's a direction that evolves and everybody moves together in a certain direction. And something similar can be observed in bacterial suspension. So these are experiments from the group of Ray Goldstein in Cambridge. And again, you can see that without the application of an external gradient, movement appears and everybody rotates in the same direction. Then also something which would be completely forbidden in classical thermodynamics, if you put some kind of well-designed objects in a bath of bacteria, like in an active bath, you can actually extract work from the bath and you can make like these small gears rotate around. And another observation is that in some of these systems you can actually create a system where the viscosity vanishes. So in these systems adding particles instead of increasing the viscosity as it would happen if you add just a particle to a fluid, you can actually decrease the viscosity. So you see that due to the fact that you have these systems that are inherently out of equilibrium, there's a number of very intriguing properties that can be observed. And this created a lot of interest in the field. But it's not only these fundamentally interesting and challenging topics, but it's also, of course, a field which has many applications in nature and in biomedicine and industrial applications. So if we think about bacteria, for example, they can swim, but they also form biofilms. Biofilms is, for example, things that we have on our teeth. It's a place where we really would like to avoid the formation of biofilms. Bacteria are transported in the soil, in the clouds, in the oceans. If you think about green energy, algae, for example, are used to produce fuel. And then there's a lot of implications in medicine. For example, you would like to avoid that bacteria contaminate, for example, conducts or patients. And sometimes also people thought about making like microscopic active particles to deliver things in the body somewhere. So in all of these systems, to understand exactly and to control the transport of these active particles, you would like to understand their interaction with their surrounding and with flows, but due to the complexity in the applications, it's actually quite difficult to fully understand these systems. So typical approaches that we do in our group is that we try to define a model system which is as simple and well-defined as possible, and also try to have well-defined surroundings and to understand some basic questions. So what I would like to present to you today is, well, the protagonist that we use in the group, which is an E. coli bacteria, and I will put this bacteria under different conditions. I will tell you what it does if I put it under flow, what happens if it gets in contact with a boundary, and also what happens if it's in contact or in the surrounding of other bacteria. And so before going there, let me say some words about these E. coli bacteria. So what you can see here is actually an E. coli bacteria that we look at under the microscope. So what you see in green is the body, and then you see this bundle of flagella, and you see that they rotate and they push the bacteria around. And maybe at some point, I never know exactly when it will happen on the film, what you can, well, maybe you saw it, there was one flagella that actually left the bundle, and at the moment where this flagella left the bundle, there was a small reorientation and the bacteria changed direction. So why do these microorganisms, these bacteria, look like they do, and why do they propulse themselves in the following way? So maybe there's something I should really point out, because it's very important, is the size. So you see a scale bar here. So the head of the bacteria is like 1 or 2 micrometers, and the flagella bundle is like 10 micrometers in length. So if we want to understand how these organisms swim, if we take into account their tiny size, we know that they actually swim at small Reynolds numbers. So that means they are in a surrounding where everything is completely dominated by the viscosity. And this actually leads to a number of constraints. So it's like if we would swim in honey, something which is absolutely viscous. That means if I stop the movement, I will stop immediately. It's very different. If we swim, we use inertia. We make a movement, we wait, we will still be transported in the flow. So if you have to evolve in these surroundings that are completely dominated by viscosity, there's a number of things. So first of all, a reciprocal movement will not lead to swimming. If you just open your arms and close your arms, you will just go back and forth. And also, if you look more at the equations, it actually means that you have to fulfill a zero force condition. So there's no force, external force applied on the particle, and a zero torque condition. So the bacteria found a specific way to answer to these constraints. And this is actually very important consequences on their shape and the way they move. So the way that the E. coli bacteria resolved this problem is actually the E. coli bacteria has this bundle of flagella that you could see, and actually these flagella are chiral, and actually the chirality breaks the symmetry. So rotating the chiral bundle will actually propulse the bacteria forwards. So the rotation of the bundle will actually create a thrust, which is compensated by the viscous friction on the head. So this is why we say they swim without an external force. Okay, one force pushes and the other one, resists. And also, it cannot just rotate the bundle. If it wants to rotate the bundle, it has to rotate the head in the opposite direction, because it swims with a zero torque condition. So put all of this together, we end with the E. coli bacteria with the typical sizes I told you, like a micron for the head, 10 micrometers for this tribal flagella bundle, and a certain frequency of rotation of the head and of the tail. And with this, they can swim up to 20 micrometers per second, so which is quite fast if you think about the small size they have. But let me make a small interlude. So I told you that we have these flagella bundle, or let's just imagine one flagella. So these flagella for the bacteria are actually rigid. So they're rigid tribal objects, a little bit like a corkscrew. And so for a long time, people were very skeptical that you can create an object where you have a body and then you attach the flagella, because you need a motor. So somehow this object has to turn. And actually, many times later, Berg, is one of the gurus that looked at E. coli bacteria, could obtain a picture of this motor. So this is 45 nanometers. So they could prove that indeed there exists a motor on this scale which connects the body to the helical flagella, and which is really like a mechanical classical rotor and stator motor. And so there's a sketch of it. So what you can see, that you have the stator and you have the rotor that turns, which will actually rotate the flagella, and it's all powered by protons. So it's like an amazing object of bioengineering that actually created this small motor which linked the flagella to the bacteria. So with this, we can understand that bacteria can propulse themselves, but we don't understand yet how can they ever change direction. And what I showed you on the first image was that on the film, that the bundle rotates, propulsion occurs. At some stage, a flagella leaves the bundle, and at this moment, we call this a tumble. The bacteria will reorient until the flagella goes back onto the bundle. And so how does the flagella leave and come back to the bundle? So all flagella have exactly the same geometry and the same chirality. So when they all rotate in the good direction, they will actually bundle together by hydrodynamic interactions. However, if one of them starts to rotate in the other direction, these hydrodynamic interactions become repulsive, and these flagella will actually leave the bundle. And so the only thing that the bacteria controls is how often the motor, or one of the motors, I don't know what I lost, but I think it will be fine. So the only thing, don't worry, the only thing that the bacteria, well, controls, not actively controls, but controls with a biochemical process, is how often one of the flagella will change orientation direction. And so if you go back to the snapshots here, you can see here the bundle is formed. Everybody rotates in the same direction. Then you can see here one flagella will start to rotate in another direction. It will leave the bundle. The bacteria tumbles until the flagella goes back into the bundle. And that's a very fundamental mechanism to explore space. So as long as the bacteria is very happy, it will try to tumble a lot, because then it will actually locally stay where it is. When the bacteria is looking for food, like for oxygen or any other kind of chemicals, it will actually have very long parts where it swims straight until it tumbles, until it finds an area where things are interesting.
What is CCW?
Oh, sorry, counterclockwise and clockwise.
Okay.
Yeah, sorry.
Yes.
Sorry. So in general, please interrupt me. I'm very happy to be interrupted. The imaging technique, it will come in a second, okay? So yeah, if there's anything else. So I'm very happy to be interrupted, and if I don't tell you everything I have on my slides, it's also not a problem. Yeah. So yeah, I will come back to the imaging technique in a second. Right now, you can just say that it's actually, it's fluorescently stained, right? So the body is fluorescently stained in one color and the flagella in another. And actually, the body expresses the fluorescence itself, and we add a color that attaches itself to the flagella. Yeah. So if now you put together this run and tumble motion, you end up with something which is like a random walk in space. So let me just show you more or less how this can look like. So this is a 3D trajectory of an E. coli bacteria, and you can see straight parts and then reorientation. And so it explores space like in a run and tumble motion. Okay, so with what we've learned so far, you see that E. coli bacteria are very specific microorganisms. They explore the space in a certain way. They have a certain swimming velocity. They can change direction with a run and tumble motion. There's always noise which is present in the system. They're tiny, so they're between a micron and 10 microns, depending on what part of the body you take into account. So there will always be rotational noise. So even without a tumble, they will reorient slightly over time. You can also consider the tumble motion as noise. And we also saw that they have a quite complex shape. So if you look at them from a little bit further away, it's like an elongated object, like a fiber, but they have a chiral flagella, and they have a kind of spherical head. And also, which I didn't really go into details, due to the fact that they have to swim force-free and torque-free, they create a specific disturbance flow around themselves. So, for example, it's like if they would eject fluid along their length, which comes from the thrust from the flagella and the resistance from the body. So they eject fluid in this direction, and they pull it back from the sides. And then there's higher moments to this disturbance field due to the fact that the body and the flagella rotate in opposite directions. Now, if we put all of these characteristics together, the questions I would like to ask: how do these properties influence how bacteria will be transported in flows, interact with surfaces, and potentially with each other? And so, yeah, that's 3D. Let me start with how bacteria are actually transported under flow. So it's a very simple microfluidic experiment. So the way that we control the surroundings and the flow is that we use microfluidics, which again flows at very small scales, at small Reynolds numbers, and we can perfectly control the flow just by designing a geometry of the microfluidic channel. So in this very simple microfluidic channel, we will inject our bacteria suspension from here, And then the channel, I think there should be, oh, this here, a cross section is just a square channel. It's quite wide and not very high. And so if you create flow in these square channels, what you have is that you have a typically Poiseuille flow profile. So the channel looks like this, and the Poiseuille flow will develop in the height of the channel. And what we are aiming at to see how are bacteria transported in this Poiseuille flow profile. And so these kind of profiles are interesting in the sense that they actually, well, somehow have a symmetry in the middle of the channel. So the velocity is 0 at the top and at the bottom part, is maximum in the middle, but the shear is maximum at the plates and is 0 in the middle. So we would like to understand how bacteria are transported in these flows. So now if you want to understand how bacteria are transported in shear flows, there's actually two components to it. First of all, I have an object, even if it were a passive object, that will interact with the flow. So if I imagine I put a fiber on the flow, this fiber will tumble while it's transported in the flow. But it will always stay transported with its center of mass along the streamline in the flow. However, if I now have an object that has a self-propulsion velocity, the orientation becomes crucial because I will swim in the direction of my orientation. So you can imagine that for a bacteria, actually the orientation dynamics will fully determine the way the trajectory will look like in the flow. And so if I look at an individual trajectory, it's like the instantaneous orientation. If I look at global transport velocities, what I want to know is orientation distributions. So this conceptually is all very nice, but how do I observe? And this comes closer to your question about how do we do the observation under the microscope. Our problem now is that the bacteria will not only be transported by the flow, but it will also swim in a direction that we don't know a priori. So in the group, we designed a Lagrangian tracking system together with Thierry Dernige, an engineer in the group, where actually we program the microscope stage in such a way that it follows the bacteria. So here you see an image where we have one body of a bacteria which is fluorescently labeled. And so it's quite easy to follow the bacteria in X and Y. You just have to take care of the fact that it stays in the center of the image. It's a bit more complicated to do this in the Z direction because for knowing where you are in the Z direction, you have to be well as focused as you can. But if you're out of focus, you don't necessarily know if you're above or below. So typically you need two images to compare if you're moving in the good direction or in the bad direction. I have to say that this over the last years has improved enormously because we do a lot of AI motivated image treatment, and now we can train the system, and very often the system already knows by looking in the interference rings, for example, if you are above or below the focal plane. So this became even much more performant. But let me show you how this looks like. So what you will see here is a film where we track one of these bacteria. Only the body is fluorescently labeled. So why do we only label the body fluorescently? Because if I have something very elongated and I'm just looking at a projection, actually this projection changes shape a lot depending on the orientation of the filament. So this is much more complicated for the image treatment program. So we are just looking at the body because it's more or less spherical. And so let me just start the film. So you will see that we are following this bacteria. Right now it's at the bottom plate or at the top plate, and you see other dead bacteria which are stuck at the glass slide. You see it perform something like a circle, and at one point it will throw itself into the bulk. And at this point you will see that the other bacteria get out of focus. Then we follow the bacteria while it swims through the bulk, and you will see here it will actually reattach at the other side, at the glass slide again, and will start to perform something like a circle again. And you see, even if there's many bacteria around, somehow we managed to follow it quite nicely. So this was nice, and we did many things with it, but somehow we were a little bit frustrated because we couldn't see the flagella. And now comes the question of the two colors. So together with colleagues from the Edinburgh University, we developed this two-color strain of bacteria, which has the possibility now that we can actually separate with a beam splitter the green and the red color. So what we do is that we track exactly as before on the body, but we have an image of the flagella at the same time. And so afterwards we can just put the two together, and then you end up with the nice film that you saw, where we can now follow in 3D a bacteria and can have also information about the conformation of the bundle.
So here the field of view goes to the Z direction
Yes.
and to the X one.
Yes, yes. So the stage displaces itself in all directions exactly, X, Y, and Z, and can now actually follow bacteria quite nicely, also with quite high speeds, well, in all directions.
And there's no flow.
So there's no flow yet, but now we'll show you the result with the flow, and we can actually also follow under flow. Yeah.
Are there interactions between
The different bacteria?
Yeah.
Yeah, so right now what I will present you is where we are always in a situation where actually that's so dilute that we only look at one bacteria. And if I reach the last part, which I hope, I will tell you how they interact. So the conditions on being dilute are actually quite strong because this disturbance flow that the bacteria create is very long ranged. So bacteria will feel the flow from the other bacteria quite far away. But right now I will only look at one bacteria. Okay. There's more you can. Okay, so the first thing we did is we put this bacteria in the channel as just showed you. So the bacteria will now be transported in this Poiseuille flow. And we look at the trajectory, so flow goes in this direction and the Poiseuille flow is in this direction. And what you see is that we have these interesting swinging trajectories, go from one side, change curvature, and go back and forth. So we thought, okay, how can we explain this? Maybe there's a very simple model we can use. And actually, there's a very simple model, not necessarily very simple, but a very well-known model, which actually tells us how fibers, so elongated objects, just an object with a certain aspect ratio, actually tumbles if it's transported in a shear flow. So this is called a Jeffrey orbit. So I have here the equations, but basically what you can imagine is I have a shear flow, so it means the velocity is higher on top than on bottom of the object. So it will actually rotate with the shear flow, but as this object is very elongated, it wants to be aligned with the flow. So it stays most of its time aligned with the flow, and then it does a quick flip and go around, and does a quick flick and go around. And now if you imagine the same thing where it's not perfectly aligned with the flow direction, it does something like a kayaking motion where it actually tumbles with the flow. And so this is very well known, and this can be cast in equations where basically I have two angles, an in-plane and out-of-plane angle, and the only parameter is this value g here, which is something like the aspect ratio of the fiber, which of course in the case of a bacteria probably some kind of an effective aspect ratio, because in reality the bacteria is a bit more complicated in shape. And everything is proportional to the shear rate that I apply. So now the very simple model is that you do something that we call an active Jeffrey orbit. I use exactly the dynamics if it were a passive fiber, and I add a swimming velocity on top. And this has been done actually by Andreas Zöttl. I think the reference was a bit higher up, maybe 5 or 10 years before we looked at it. And so the only parameters that you have to adjust is the effective aspect ratio, and then you have the swimming velocity of the bacteria. So before going there, I just want to show you a quick simulations on how such a Jeffrey orbit looks like. So what you see there is this kayaking motion I told you about. And so this was the rod that did like this one loop. So you can imagine that the tip of the rod goes around on a unit sphere. And then this is the kind of the phase space that is folded up. And so you will always go around the same trajectory on the unit sphere. And where you will do your orbit depends entirely on your initial starting position. Okay. And also something important to keep in mind, if I have a rod, if one, well, the rod doesn't really have a head, but if the head points in one direction, it will always stay on this direction. There's no way it could flip on the other side. Yeah?
How expected his behavior depends on the height of the channel?
So the height of the channel, it mainly defines the shear rate. No, it does. So the shear rate actually enters, can I go back? I don't know. Yeah. So the shear rate enters here, okay? So right now it's a local picture that I look at the local shear rate, but then it will swim and explore different, yes, absolutely. Oh, okay. Okay, so now if we think about putting this together, we start to understand why the trajectory looks like this. So when the bacteria starts to leave the surface here, it's in the bottom part of the Poiseuille flow. So in the bottom part of the Poiseuille flow, the shear rate has a certain sign, and in the top part, the shear rate has another sign. It means here, things want to rotate in this direction, right? The flow velocity is higher here than there, so the rotation works like this. So the bacteria leave the surface, and they're reoriented by the Jeffrey orbit. And at the same time, they swim. But interestingly, when they cross the midplane, so the midplane is the plane where the shear rate is 0, and then the curvature of the flow goes in the opposite direction, and so the rotation will also change direction. So the fiber will be oriented with this rotation direction, and once it crosses the middle, it will be reoriented in the other direction. So this can explain these swimming trajectories, only putting the dynamics of a rigid rod with the swimming velocity. And now if we look in more detail, we can actually see that we have a whole family, and I think this is somehow what you said about the height of the channel. The control parameter is the ratio of the swimming velocities of the bacteria and the flow velocity. And so you can recover these swinging trajectories. If you have a very high shear rate, then actually the bacteria tumbles so strongly that it cannot leave the quadrant of the flow. And some of them are actually, oh sorry, I don't fully master the arrows on the thing. And some of them, they leave the surface and they're just reattached right away to the surface, and some cross from one side on the other. And this is actually a very nice agreement of simulations of data. So this was a first look at these systems. It's really explained by a quite simple picture. But first of all, I only showed you things where the bacteria were nice and actually were evolving in the plane of the shear. So of course, the bacteria can also point outside the plane of the shear. And so now this is a trajectory from the simulation where the bacteria tumbles, like in the kayaking motion, and while tumbling, it swims towards the side. Okay, so what you will see on the film is that they swim towards the side with this oscillatory movement, which we can just fully explain by the fact that they have a certain drift velocity because they're oriented towards the side, and then they just oscillate around. And we can also observe this from the experiment. But it still seems like a really simple model. So first of all, we know that there's noise in the system, so why doesn't noise play a role? And also we know that the bacteria really have a complex shape. So let's look in a bit more detail what happens if I look at longer times, because if you look carefully, all experimental conditions I showed you is like 1 or 2 tumbles and not at all very long trajectories. So the next thing that we want to look at is actually, does this chirality of the flagella play a role? And indeed, people have observed that when I put bacteria in a shear flow, that somehow bacteria don't follow the shear flow, but they seem to have a tendency to swim to a certain direction. I would just say towards the right, but basically they orient within the vorticity direction. And this has been explained again by a purely hydrodynamic effect. So right now, in whatever I tell you is never something biological or physico-chemical, it's just really the shape of the bacteria that interacts with the flow. And the rheotactic effect can be explained in the following. So people have shown that if I put a gyral helix under flow, that this actually helix wants to drift towards the vorticity direction. And now you can imagine that this, so now we look from above, flow goes in this direction, the helix wants to drift in one direction, but the head, it is a bit like an anchor. The head is much larger than the flagella, has a lot of viscous friction, so actually the anchor does not really want to move, so what we create is a torque. So it's called a rheotactic torque. It means that actually if I have head and flagella, then actually the flagella want to rotate around to be, as much on the side as possible, and the flagella want the object to be aligned in the vorticity direction. So now we would like to understand a bit more how these dynamics come along. And so we use exactly the same setup as before. We use again this Haley-Shaw cell. We inject bacterial suspension in it in a very dilute regime. And now what we can do is that we will actually scan through the height of the channel and observe bacteria at different levels, close to the surface or in the bulk. So the way we look at things now is that we want to get statistics because I want to know what happens at long times. And so if I show you these first 2 films, so these are bacterial trajectories close to the bottom and close to the top. And I hope you see that those somehow go, to the left, and those go towards the right. So they do not just go straight. And the bias in the transport is actually opposite if I'm at top and then bottom. And this again comes from the fact that the shear rate changes sign in the middle of the channel. So how can we explain this? So the first thing is that we measured a bit more in detail these velocities. So what we did is that you have the transport velocity, and so we measured the component, only the component perpendicular to this velocity, which we call the drift velocity. And so this is what is represented here as a function of the channel height. So what you can see is in the middle of the channel, there's no induced velocity towards the side, but also in the middle of the channel, the shear rate is 0, so that makes sense. If I go closer to the wall, this induced velocity gets higher. It also is higher for a higher flow rate. Again, in agreement with the idea that the shear increases if I go to the wall. And we also see that it changes sign if I go from the bottom to the top. And very importantly, we checked with passive particles. So if I just put a passive tracer in there, this tracer is transported completely straight and has no velocity component towards the side. And we also needed that to perfectly align the microfluidic channel, because later on we want to measure orientation distributions. So we really have to know that we are perfectly aligned with the flow. And so from this, we can actually rescale things as a function of the shear rate. So what is plotted here now is this drift velocity, so only the velocity component perpendicular to the flow velocity, and it's normalized by the swimming velocity of the bacteria. So there's two things you can remark on this curve. So first of all, well, whatever I do to the system, it always scales on the shear rate, and there's also some simulations which I will come to later. But first of all, it increases linearly in the beginning with the shear rate, and it seems to saturate, but very slowly. But from the simple picture I told you, we would just expect that the rheotactic torque would put the flagella on one side and that everything should want to be perpendicular to the flow. So perpendicular to the flow would mean like 1 here, so we're actually quite far away from perpendicular to the flow. And also somehow we are missing another ingredient. Why would things increase linearly with the shear rate? So maybe there's some time scale or something we're missing in the system. So to understand this a little bit better, we wanted to look in more details on what we had, because what I show you here is just the average. Oh, I'm really not in control of this. What I show you here is just the average velocity, but there is a distribution to this velocity. So we looked at the velocity distribution, and what you can see here is that if I have 0 shear rate, the velocity distribution is flat. Then if I increase the shear rate, the velocity gets more and more biased towards the vorticity direction, but you can see that the distribution stays quite large. And so we know that velocity actually comes from orientation, so we also measure the orientation distribution. And so here you see the orientation distribution of the bacteria, and again at a 0 shear rate it's flat. It becomes to be biased towards the side, but what you can see is that there's not a single peak that evolves, but there's actually 2 peaks, and these peaks get more pronounced and maybe move together when I increase the shear rate. So what that means is that bacteria are not perpendicular with the flow, but a little bit above and a little bit below. Okay? So I do not see the signature of these 2 peaks in the velocity, because the projection of these 2 velocities on the perpendicular direction is the same. So let's try to understand this, and to try to understand this, we can back to the simulation results. So we use again the Jeffrey orbit, and we put additionally on the Jeffrey orbit something that describes his rheotactic torque.
Sorry,
Yeah?
it should be symmetrical.
It,
somehow in
yes,
but I don't see any symmetry here. On the
symmetric with respect to.
we should have the opposite angle.
So what so I'm not sure if this drawing isn't, so the 0 is the flow direction, okay, and pi half would be the vorticity direction, okay? So the flow direction, the vorticity direction, and so we know the bacteria want to go towards the vorticity direction, so the rheotactic torque breaks symmetry, right? Because of the chirality. So if I had, if the flagella had a different chirality, it would go in the other direction. Yeah, exactly. No, it's a very important point. So all of this comes from the fact that the flagella are chiral. So if there was no chirality, I could not break the symmetry. Yeah. Okay, so this can be added as a term to these equations, and so you add to the Jeffrey equations, again taking into account orientation dynamics over the 2 angles. You add here a rheotactic term with something which we call nu, which is a new parameter that we can adjust, which is this rheotactic strength. And later on we will also add orientation and noise. But let me just show quickly what this rheotactic term does. So as a reminder, oh, nearly, as a reminder, okay, let me try again. Okay, as a reminder, this is Jeffrey. Okay, if I only have Jeffrey, I always stay on the same side, I only stay on the same trajectory. But now if to this I add this rheotactic term, then you will see that actually if the head is on the wrong side, that I will spiral outwards, my amplitude will increase until I cross at some stage the flow plane, and I will actually orient the head on the other side. And once the head is on the other side, I will spiral inwards to reach something which is as close as possible perpendicular to the flow. Okay? So it's very different from a Jeffrey orbit. It's really a system which now evolves towards a new stable position, which is this perpendicular orientation. But you could also see already that this spiraling inwards is actually quite slow. So if you put all of this together and now we simulate these equations and we add rotation and noise to it, I will now show you how these orientation distributions look like. So bear with me, this transparency is a little bit full. So what you can see here is again these phase spaces that we knew from the Jeffrey orbit. And on top, the color tells you the orientation distribution. If the color is bright, it means a lot of particles are oriented in this direction. So this one is without the triviality effect. This is just typically Jeffrey orbit. And basically the only thing you see is what I told you in the beginning. Things want to be aligned with the flow. And as just in the Jeffrey, there's no head or tail. We have most of the fibers, if I increase the shear rate, are oriented upstream or downstream. So the noise is very important here and much less important here, because the noise, we should describe it as a Peckley number, as a competition between the rotational noise and the shear rate. Okay? So here things are smeared out, and here clearly things are aligned with the flow. Now I add this torque to the system, and we see that immediately things are quite different. And very soon things are oriented towards the right side. But what we can also see is that somehow with a lot of noise, we already still have like a little bit of a preferred direction downstream or upstream. And this existence of the two peaks is actually that the upstream and the downstream slowly bend towards the right side. And actually it's these two peaks that come together until maybe one day they merge and will be perpendicular to the flow. Okay? So we understand better why in our orientation distribution we have these two peaks. But we can also do more. We can actually analyze these different positions, and we can start by looking at what we think is a fixed point. This is being perpendicular to the flow, and it actually turns out that this fixed point is only marginally stable. Marginally stable means that you will circle inwards for an infinitively long time without never reaching it. So this explains us why, if we looked at the drift velocity, it wasn't at 1, because it takes a very, very long time for bacteria to orient really perpendicular to the flow. And what we can also do is that we can actually analyze these 2 points, and we can see how they evolve if I add this rheotactic drift towards the system. And we can actually see that these 2 points will linearly converge together. And this is actually what explains the linear increase of our curve. So just to show you this curve again, the control parameter is actually the strength of the rheotactic drift, the strength of the rheotactic torque, which is this rheotactic coefficient and the shear rate, and it competes with the rotation and noise. So the time scale that was missing is actually the rotation and noise. So this is why we have this linear increase in the beginning, and then we know that the stable point is only marginally stable, and so this is why actually we drift very slowly towards this plateau value. Okay. So by looking into the system in more detail, we can describe how they evolve under flow. And again, this only takes into account the shape of the bacteria. And this actually motivated us to make another test, and so I will make a small interlude. So working with bacteria in our case is we very often use them like a model system. So right now I describe them to you as an object with a strange shape. But we have absolutely no control on this shape. So the nice thing about the biological system is that it reproduces itself with exactly the same characteristics over and over. But if I want to change anything, this is actually very hard. So we thought maybe we can do something like a passive bacteria. And I'm very happy to say that I'm making passive bacteria because many people make active colloids, so we tried to make a passive bacteria. And so nowadays we can actually microprint things on the micrometer scale. So this has been done using a nanoscribe. So the nanoscribe is really like a 3D printer. It prints inside the bulk of a resin. It's a 2-photon technique. So actually only where the laser is in focus, the light intensity is strong enough to induce the cross-linking reaction. And so you move through the bulk and you can produce your object. And the resolution of the voxel is typically like a micron in height and some nanometers in width. So you can indeed produce these passive bacteria, which are a little bit larger. So the head is like 10 microns and the tail is maybe 30 microns. So now we have full control over the shape. And also we have a system that doesn't have noise. So we can look individually really if this geotactic drift exists or not. And so what we did is that we changed the pitch, and also we could produce something where we knock out chirality completely. So we produce one helix that has a chirality in one direction and then one that has chirality in the opposite direction. And so once we have them, we have to bring them in the channel. And what you can see here is that actually we have a micropipette that we bring close here. It's only a helix to a helix that was attached through the printing process. So we detach it, and then we actually suck in liquid, and then we can grab the helix in this way, and then we can bring the helix in a pool. We eject the helix in the pool, and then we can look at the helix under flow. And so if I do this now for 2 helices, and now I have changed chirality, so now they should explain the symmetry break-in, you see that they do something very comparable. You see these fast dynamics of the Jeffrey orbit. You see a slow rearrangement process, and we see that in the end, the 2 chiralities will indeed lead to an orientation in different directions. So with this, we are really convinced that this rheotactic reorientation fully comes from the shape of the object. And it's also something that we can use if we want to direct things, orient things in the way that we want, if we microprint small objects. And so let me just quickly see where I am. Okay, yeah.
This is obvious, but in the bacterial case, since they are active,
Yes.
why don't they just orient with the flow?
Oh, so the bacteria orient. So the bacteria has always a swimming velocity in the direction of its orientation. However, in the direction, the orientation is given entirely by the interaction of the bacteria with the flow. So the bacteria cannot decide I want to swim to the left or to the right. The bacteria can just swim straight. So they will spend a lot of time aligned with the flow. But if they have this chirality from the flagella, on top of it, it will actually reorient them towards the vorticity direction. Yeah. So they cannot decide where they want to go, basically. The only thing that they can do, they can run and tumble. Yeah.
To follow up on this, so it means that the effect you see has nothing to do with activity?
No. So
These particles at rest.
no, I agree. So somehow the picture is really that up to a certain extent at last, you can treat them as fully passive particles, probably with noise. And then once you have the orientation distribution, you add swimming velocity on top, and that's everything you need.
And it also implies that the run and tumble, and particularly the tumbling reorientation, don't really affect the
Yeah.
structure of the...
So I didn't say that. So it does. So in many cases, you can just cast the tumbling into the noise, and it increases the rotational diffusion coefficient. It's not exactly that, but mostly it's what you see. Yeah. Okay.
And I guess you're in a very non-dense regime.
Yes.
Would you keep artificially? Or?
So we are, I would say, in a very artificial regime anyway. So we are very dilute, so we can look at one object, but also we make the bacteria happy enough to swim, not happy enough to divide. And also the fact that we keep them happy enough and we have enough oxygen everywhere, there's no gradients, there's no oxygenotactic or chemotaxic. So we try to be really in a regime where we can, the only thing that will act on the bacteria is the flow. And we also treat surfaces in a way that they don't attach and don't form a biofilm.
And if it was a bit denser, would they try to use some collective?
So if I still have a bit of time, I will maybe jump to the collective motion. Okay. So So if one day you want to hear about the surface, I'm happy to talk about the surface. But now I think it's indeed really nice to think what happens if I have a bacteria and another bacteria not too close. So the interesting thing to keep in mind is that bacteria will actually interact with each other hydrodynamically, but they are still very far away. So we can have 1% or 2% or up to maybe 10% of bacteria, which is still really dilute, but they will start to feel each other. And they will actually feel each other because this is a schematic of the flow that I told you. One bacteria will actually induce this disturbance flow. And this disturbance flow looks like this. It pushes fluid outwards there, and it actually pulls fluid inwards from the side. And this disturbance flow is very long range. So if I have one bacteria swimming and another bacteria swimming next to it, it will actually feel the flow. And if you look at the symmetry of this flow, you can understand that they actually want to align with each other and swim in the same direction. So people have made models about this, and here's just a number of them, and they have proven theoretically that collective motion should occur, meaning that there's order, that they will actually align in the same direction, and that these hydrodynamic interactions would lead to some kind of a long wavelength instability. And indeed, this is what you can see in the experiments. So this is an experiment of E. coli bacteria. So what you can see with the red arrows is that there's this collective motion that appears. And I want to point out that this is 100 micrometer the scale. The bacteria are 2 microns. So what you see here as a movement is something that happens on a scale which is like hundreds of times larger than the individual bacteria. So this was all very well known, but somehow the question remained if there's an intrinsic length scale that would be selected. So these kind of vortices that you see, would they always be formed on the same scale? And what would that scale be? Because we know what aligns them together, but there's nothing that opposes itself to this alignment. So we wanted to understand this question more. So we actually created just a reservoir where we could put the bacteria. We are very, very careful that there's enough oxygen from everywhere. And what we do is that we vary bacteria concentration, and we vary very strongly the height of the channel. Because if there's an intrinsic length scale, maybe at some stage, if I make a very huge container, I will suddenly see that I have vortices that are smaller than the container size. And so what we do is that we mix bacteria and tracer particles, and we just follow the movement of the tracer particles. And so the first thing we had to find out is at what concentration do I have this onset of collective motion. And so here you have a number of pictures, and you see volume fractions are very, very small. If you have very tiny volume fractions, there's kind of a random motion, and then somehow you will start to have collective things here like streaks and here like wavelengths. But it's not so easy just by looking at it to know if this was the onset of collective motion or not. So what we did is that we calculated something which we call the velocity correlation function. So it means that you stay at a certain position, you look at the fluctuations of the velocity over time, and you calculate the decorrelation function. And from this you get a certain time scale of your system, and what you can actually show if you vary the concentration, that this time scale diverges, well, diverges capped, but gets very high, and then decreases again. And this is a bit reminiscent of what we have in a second order phase transition. So we decided we take this concentration where the time scale of the system is the highest and say this is the onset of collective motion. Mm-hmm. And so using this, we can actually indeed, for many, 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 many experiments, have a phase diagram where I have on the one side the volume fraction and on the other side the height. Because in the system there are two things that can stabilize. It's the noise in the system and the confinement. And indeed what you see, and maybe I have a line for this, what you see that the critical volume fraction actually scales with 1 over the height. If your chamber is high, then you have an onset that happens earlier than if you're very confined. So this is in perfect agreement with people who model like kinetic theory of this onset, but there's something which remains quite surprising, is that actually we never saw a saturation. Because I tell you, if I put them very far away, they would still feed each other, but we have noise in the system. So there should somehow be a concentration that is so small that the noise will actually overcome the alignment. So this, for the moment, is still a mystery, what happens with the noise in the system. But what we can do, we can now exactly define where's the onset of the collective motion. And once I have the onset of collective motion, I can now for every height in my system, go to the onset of collective motion and look at the patterns that evolve. And here are some pictures of these patterns, and we actually have scaled them with the height of the system. And what we see, even if we go to very, very high heights, which get close to 1 millimeter, which is a lot for a bacterial system, the scale of this collective motion always scales with the height. And this is actually what I show here, and maybe I will accelerate a little bit. So it just means that there's no intrinsic scale that we can find in the system. And if this was not convincing enough for you, I will show you one last thing and then I will be done. Right now, what we saw is whatever we do with the container, the length scale that emerges is the height of the system, which is the smallest scale in the system. But there's another scale, which is the radius of the system. And indeed, what we could observe, to our big surprise, I have to say, is sometimes we have these enormous vortices that developed and that fill the full container. So clearly here, the scale of the system that dominates is the width of the container. And they are so stable in time that actually what you can see here is that actually Benjamin stitched together different observations because it's so big that actually we cannot observe it under the microscope. And so we have these very, very persistent, huge vortices that develop. So again, here, the wavelengths of what we see scales with the system size. So it's very clear from these results that actually active turbulence is scale-free up to several millimeters. So now this poses important constraints for theory and is hopefully a challenge to find out exactly how this is. And just the last thing, both of these states, the large vortex is unstable and can actually break down into these smaller vortices, and sometimes less often these smaller vortices coarsen and form this huge vortex. And there's more things to be done. Fancy things that we're doing right now is that we follow an active tracer through the bacterial bath, and so you can locally have access to the properties of the turbulent motion, because around this tracer particle we see the dynamics of the bacteria, so there's much more things to be done. And I will stop with this because I think I've already been a bit long.